Our final presentation for today comes from Nicholas Nystrom, CTO at Peptilogics, where he leads development of the Nautilus generative AI platform for peptide drug design. Prior to joining Peptilogics, Nick was chief scientist at the Pittsburgh Supercomputing Center, where he architected the first supercomputer to converge AI and HPC. For today, he's going to be unlocking chemical space for drug design with generative AI. Nick will provide an overview of generative AI methods for addressing the high dimensionality of chemical space and the complexity of biology, together with HPC simulation methods to elucidate the resulting molecular biophysics and illustrated by results for generating peptides with properties that surpass those of human-designed peptides. Nick, welcome to the conference. How are you today?
Hey, Brian, thanks for the introduction. Thanks to everybody for having your attention this late in the day. And it's a pleasure to be back at the HPC AI Advisory Council. So as Brian introduced, I'm going to be talking to you today about the shift. The first talks today were fantastic. They were hard act to follow. But I'm going to talk about another grand challenge, and that is developing new therapeutics. And this is especially important because, as we are all well aware, the cost of healthcare is going up. But what is less obvious is that the cost of developing new therapeutics, in terms of the cost, the risk, and the time, is also going up exponentially. And so that's what we're addressing at Peptilogics. Some legal disclaimers and a very brief introduction. So Peptilogics is a small biotech. We're headquartered in Pittsburgh.
One sec, Nick. I'm sorry. Are you sharing something? Because right now I don't see a presentation.
Excuse me. Yeah, this is I traditionally forget to click the share button once I specify what to share on Zoom. Are you seeing it now, Brian?
Yes. Perfect.
Okay,
thank you, Nick.
Great.
Sorry.
Well, let me just get the other window out of the way. Okay, still look good?
Yes.
Okay, fantastic. Thank you for letting me know. So we're a small biotech. We've been using HPC and AI extensively from the outset. I joined the company in 2021. We built our own on-prem supercomputing. And as many of you know who knew me from before, I was previously at the Pittsburgh Supercomputing Center. And what we're building at Peptilogics really builds on those learnings and takes them a step further. So first, why are we doing this? The two charts on this page show the rising cost of drug development. Each of the four bars is a different decade. And so we can see is they're roughly doubling every decade. And over half of the costs are in clinical development, but a large amount is in pre-human. Coming from a research background, as many of you are, I think of numbers like $1 billion as a lot of money. But the fact is, in therapeutic development, these are table stakes. This $1 billion, or 1.1 billion, is the pre-human clinical cost to develop one drug. And obviously, we have many untreated diseases. There are, by different estimates, between 6,400 and 10,000 rare diseases currently in the world among humans, along with non-rare diseases. We have approved therapeutics only for a small number of those. And so this is increasing exponentially. At the same time, on the right, we see a chart from IQVIA Institute looking at the productivity of clinical trials. And these are going down over time. And they measure it, the reports referenced, but they measure it in terms of success, complexity, and duration. And a lot of the reason for this is that the low-hanging fruit of medicine, of therapeutic development, has been plucked. In other words, the things that have been easy to find have been found. And what we're facing now are much harder challenges biologically and chemically. Now, as many of you know, biology is messy. As hard as physics is for NIF or seismic exploration is for petroblasts, biology is inherently noisy. We can't explore everything we want to. We can't measure things precisely. And those measurements are very time-consuming, costly, and imprecise. So we're looking at this from a new perspective. And that perspective is through the lens of peptides. Peptides are interesting. I want to spend a few minutes just saying what they are because I realize people who are listening in come from all different backgrounds. Traditional drugs are what we call small molecules. What we have here is a small molecule called zymaldine. And one of the challenges of small molecules is that they're not selective. They tend to bind to different targets in a biological system. It's why chemotherapies are known for being so toxic. They kill not only the cancer they're after, but they also kill other cells indiscriminately. Zymaldine was a drug that was pulled from the market decades ago because it was toxic. And this is meant to be sort of to scale. So the small cartoon where my cursor is, if you see my cursor, is meant to be to scale. I've blown it up so you can see the structure of what a small molecule looks like. And these are small. This is 0.3 kilodaltons or 300 AMUs. At the opposite end of the scale is where a lot of current research is, in what's called biologics. These are things like antibodies, RNAs, and so on. Here we have a drug that its generic name is here. Its brand name is Keytruda. It's a blockbuster drug, tremendously important for cancers. You can see it is substantially heavier. It's 149 kilodaltons. But because it is so much bigger, this also makes it harder to manufacture. It makes it more expensive. It also means it can't get into cells as like a small molecule can. Peptides are in the middle. And so peptides are, again, this is sort of to scale. Here's an alpha helix in the biologic. Here's an alpha helix in a peptide. Those diameters are roughly on scale. The points of peptides is we can manufacture them affordably and efficiently. Because they are nature's natural carriers, they have tremendous safety and selectivity. They can target exactly what we're after. We can engineer them to get into cells, to reach intracellular, even intranuclear targets. We can modify them in ways that are predictable given the right amount of training on our models. We can modulate pharmacokinetics, and we can synthesize these in ways that become very complicated to do special things. And so that's the playing field, and that's why peptides are where we're working. And because they are composed of amino acids, they're amenable to this sort of design process. It's an optimization problem. I showed this slide once before, some of you may have seen it, but it illustrates the point. The way this is done traditionally is what's called a display library, or a high throughput screening library. In that library, there might be 10 to the 12th peptide compounds. 10 to the 12th sounds like a big number that you can test in high throughput. But even if you're only comparing against the peptides of length 30 with only standard naturally occurring amino acids, that ratio of 10 to 12 versus the number of options for those peptides is almost identically to the ratio of a drop of water to the volume of the world's oceans. And so will you find what you want in this high throughput screening library of 10 to 12 compounds? The answer is probably not. And what people are finding is they usually don't. And so we're doing this optimization with AI and HPC, looking at both the design space and extensive biophysical simulation in terms of molecular dynamics and some quantum chemistry to be able to predict peptides that do we want, to design them to do specific tasks, and then to understand how they work. And we believe this to be possible why we set on this journey, because we had previously done things like, you may recall, the poker Labrada spot that achieved or surpassed the world's top humans in this challenge of unstructured information. And these spaces are similarly large. So that's where we came from. That's where we thought we would succeed. And what I'd like to tell you about today is where we are, where this has gone, and in particular, why generative AI that we're hearing all about in the press from ChatGPT and DALL-E and all of the other generative models actually apply to things in science we care deeply about. So a few words to level the playing field, since again, we all come from different backgrounds. Everyone has heard the words generative AI. often it's not discussed what that really means. So what I mean by a generative model is a statistical model that samples a probability distribution on an observable variable X and a target variable Y. It can also be unlabeled, in which case there's just X. But then once we have that probability distribution, we can sample from it and generate new instances. So for ChatGPT, that's generating new instances of text given a prompt. For image generation like DALL-E and Midjourney, again, it's give a prompt and it will give you a new image sampled on some large space that then represents what you have prompted for. And the ways that we're using this in drug design, and not just we, but also others, are generating hits. And by hits, we mean molecules that bind to a biological target to act in certain ways. For example, to suppress function that that protein would be doing, or to amplify it. Lead optimization, which means when one has a molecule that is close and one would like to make it better, to be more drug-like or more amenable to working as a therapeutic people can use reliably. Generation of molecules having specific functionality. So we'd like things to be antimicrobials. As we all hear, antimicrobial resistance is a major problem in the world, with new strains of things like gonorrhea coming up and appearing that are resistant to all known antibiotics. Saying we would like a molecule to have specific functionality and go further would be a real win. And then there are others. One that I want to hit on, though, is exploring chemical functional space. And by that, I mean not just structures. I think we can easily generate structures, but we want structure to have specific function to do things that will be beneficial and treat disease. I want to go into 3 quick examples of different kinds of generative models that are applicable to peptide design. And those are variational autoencoders, transformers, and large language models, and then these DDPMS. So first, with variational autoencoders, these have been around for a while. They were popularized from a paper from Dirk Kingma and Max Welling back in 2013. And the point here is in the center, we have an encoder, we have a decoder. We train the encoder on a set of data, X. We encode that data to a lower dimensional space. So we go from Rn to Rm when M is smaller than M. And then we have a decoder that samples that latent space to produce additional samples or additional data representing different qualities. And so if we were to train on images, we have a latent space that reduces that to, say, to dogs, cats, cars, and trains. And then we could deal with things like latent space. We'd say, show me a cat, show me a dog, show me something that's halfway between a dog and a cat. And so that's where you see a lot of these image generation programs working. But we can also do that in terms of these molecular properties like solubility, lipophilicity, antimicrobial, minimum inhibitory concentration, and so on. And so we feed in our molecular structures represented somehow. In this case, it's what's called a SMILES string, which is a serialization of that structure. A series of properties that gets encoded, we decode it, and out we get additional generated molecules with desired properties. And for the way that looks, here's an image from a recent paper by Dean et al. On something called peptide variational autoencoder, where they're looking at generating antimicrobial peptides. And here we see them with 2 different levels of effectiveness at less than 0.2 and greater than 0.2 in terms of log micromolar concentrations. A second way of generating peptides, and there are many more that I'm talking about here, are transformers. And everyone's heard about transformers. They come from a paper from Google, from Vaswani et al. On attention is all you need. The point of transformers is they let us learn long-range dependencies. And that's important in language because we want to understand the relationship between words in a text, sentences, phrases, and so on. The same applies to proteins. Remember, peptides are composed of amino acids. They're small proteins. And the way they fold, everything we hear about with structure, with AlphaFold, those bring these distant amino acids together. And so distant relationships become very important. And what we can do with these transfer models is we could train again on large data sets And these can be very large because this becomes unsupervised. And what we can produce then, autoregressively, which means a sample token after token, are additional samples or additional peptides in this case, or proteins in the figure, that have desired properties. So again, these are antimicrobials, and they're looking at 5 families of lysozymes and showing that where red are naturally occurring ones, blue are additional, you can produce many others that may have desirable properties. And that's a very recent paper from actually Salesforce and their AI team. And then the third category that I want to mention here, because it's also very interesting and very timely, are denoising diffusion probabilistic models, where here we essentially do a self-supervised learning coming from a data set, in this case, a picture of a nice little dog, doing a noising phase, learning from that, and then developing a reverse denoising. So we can essentially introduce noise, we can remove the noise, and we can generate samples then from a Gaussian distribution conditioned on certain labels. And here's an example from recent work before I get into our recent results. And this is something called this is a diffusion model on proteins. And this is looking at conditional generation using diffusion model on this one structure. These structures like 6C0B are structures in the protein data bank. And this is generating binders to this protein. And so one can do a lot with these as well. So moving forward to where we are, we have our platform, again, we're running on internal HPC systems. It's composed of multiple layers. So we've named it Nautilus, and the point of Nautilus is it implements convergent design. We put all of the properties we care about up front and do optimization on them jointly. Traditional drug discovery works by saying we want to find binders. And then from those binders, we want to look at activity. And then we want to layer on properties with absorption, metabolism, the things that make it drug-like. And the problem with that approach is it takes a long time. And at every stage, one may have what looks like a great set of binders that molecules bind very strongly to the target, but they can't be made soluble, or they can't be made cell penetrant, or they can't be made compatible with the formulation, say, for making it an oral drug instead of injected. By putting all of that up front and doing design across the joint distribution, we can eliminate those brick walls and those occasions where one has to start over, and those cost time and money. That's where that $1 billion comes from for the preclinical work. It's all the failures prior to getting to clinical status and putting these drugs into humans. So what we built, we have a large collection of data we have homogenized, curated. There are 80 plus data sets. We've named this REEF. Again, it keeps with Nautilus theme. Nautilus is a nice little cephalopod, and a reef is an underwater structure where a lot of energy and diversity comes from. So that's where a lot of our training data comes from. Training data sizes here are up to hundreds of millions of data points. And there are also others, they're down in the hundreds. And that's where it gets really interesting. I'll talk about that. We have a novel representation that we have named quiddity. And the point of that representation, think of this as analogous to something like GPT-3 in language. This is a model representation for proteins and peptides that lets us learn from less data. It lets us do transfer learning, and it supports everything on top of it. In terms of peptide generation, these are generative AI models. These are named after the deepest points in the oceans. So the 4 I'm going to mention today are Challenger, Galathea, Diamantina, and Calypso. And these are producing peptides for high affinity or high binding strength, high selectivity, in other words, binding to one target but not to others, and in different conditions. We then have a number of predictive AI models for solubility, lipophilicity, toxicity, whether or not something can get into cells, whether or not it can cross the blood-brain barrier, whether or not it's an anti-inflammatory, and so on. And we use these in conjunction. We use the generative AIs to predict molecules that bind, and the predictive models, as of today, to then rank those according to what we want, again, from the outset, for any given biological application. We then do both computational and empirical validation of those compounds. So at this stage on the right, we do a lot of simulation. And this ranges from fairly straightforward model-based biology to atomistic, long time scale molecular dynamics. And the point of that is once we get molecules that emerge from the center from our AI models, we then want to understand in detail how they interact with biological protein targets, whether they are agonists promoting activity or antagonists reducing it or something else, and where they actually bind to the target, because it turns out biology is very robust and there are many ways to do this. We then synthesize and validate those in a wet lab. And when we emerge from this, we advance to development. If we have to do more work, we iterate. A point where we're different from the models being discussed for content generation is that because we do this step of synthesis and validation, we don't worry as much about the phenomenon of hallucination, because if we do find things that are off base, we'll typically find those in simulation. If we don't find those in simulation, or if it's not conclusive, then we will certainly find them in the lab, in vitro. So an example of how this works, I want to put this in context with what people know is generative AI. So on the left, everyone knows visual models, image models, like Midjourney, DALL-E 2, Imagen, and all the rest. So in this case, it's an example of how one of our generators works. I prompted Midjourney to give me an image of two panda bears in a laboratory. It was totally bizarre, not something that would be trained on, and it did a really nice job out of the gate. What we can do with our models is analogous. So in this case, this is with our challenger model, which generates a peptide given only the target sequence, not the target structure. In this diagram, we see structure, but we don't give that. So we just get a sequence like MLA, MAS, TT, so on, where each of these letters is the code for one amino acid. And given that sequence, we can get a peptide that binds to the target, to the correct active site, and that even generates the correct activity. And so that's the power of these generative AIs in terms of getting us to things that would not have been possible previously. And the other thing we're finding is that the peptides we're generating with these are outside the experience of our human peptide scientists. In other words, these peptides are things that they say they would not have generated. They find to be biologically valid, but things that are beyond what they have worked with before. And then, of course, we just a bit more on the predictive models and the sort of biophysics and lab work we do. But to keep it moving, I want to say a few more about what these models are. And these are the generative AI models. So in the spirit of this conference, these are typically running on collections of currently Ampere class GPUs. We have built our own system. These are all NVLink connected GPUs, the highest end we can get at a given time, and we have the ability to incorporate novel hardware as we move forward. So Challenger can bind to any target, given only the sequence. And the beauty of that is it lets us look at novel targets for which nothing is known. And a recent study from 2018 from Tudor Opria's group showed that only 3% of biological targets had therapeutics that were clinically approved. The other 97% did not. And so novel targets are a large unexplored area of value for developing therapeutics. I know I'm getting low on time. Galathea is a complement. This will give it the structure and optionally where we want to bind in that structure, and then get peptides that bind to that interface. So this could be great for disrupting protein-protein interactions, addressing other areas where we think of targets as being, quote, undruggable. In our view, they are druggable. Dimensionality lets us do lead optimization. So given the scaffold or given prior knowledge, we can optimize based on that. And that's based on some additional algorithms. Each one of these classes is a different class of algorithms. And then Calypso are additional ways to do this, especially focused on expanding chemical space. I don't want to spend a lot of time on this slide, except to say that for a number of these classes of predictive models, we're achieving state-of-the-art accuracy. Again, this is pulling together the HPC, the AI, some HPC for the training data, but then really looking at going beyond what the published models do. They typically do only standard amino acids, but we're focused on going beyond that to non-standard amino acids, cyclization, and branch peptides. The workflow is here. I want to jump forward to the next slide briefly, because one of the areas I want to cover in this talk is exploring chemical space. And where that's really important is looking at function, not just sequence or structure, but what does the molecule do? And one of the other areas where we can contribute is generating these focused peptide libraries. So unlike the random screening libraries mentioned at the outset, these are focused. These are peptides they're predicted to bind. And on top of that, we have layered on a variety of properties. They're also predicted, such as solubility, hydrophobicity, isoelectric point, the things the peptide scientists and medicinal chemists care about to produce drugs that will work in people. And then based on this set, this is a UMAP plot of about 890,000 sequences, we can select. And if we had this set down in bright blue of qualities we want to make a drug, we can look for where those all co-occur and then say, okay, these are ones we want to focus on. And that's something that will really help with the developability of these compounds and driving the field forward. And where I want to end this really is on this slide. And this is what was surprising to us to some degree. And so this was out of the gate. These were early results. I want to put them up because they were cycle 1 for a certain GPCR or G protein coupled receptor target. And the key here is that sequences were designed by an experienced protein chemist. We find is, yeah, they bind. They're fine. But they bind to the target, which is V2R, as well as to two off targets, V1A and V1B, approximately equally. And the problem with that is you have off-target toxicity effects and side effects. Whereas the AI-generated sequences were not only novel, so outside what our smart people have designed, but they're also selective. They bind 3 to 4 times more strongly to the target than to the off-target. And so it tells us the generative models are not only producing high-value hits, but they're also producing hits that go beyond what traditional medchem has done. And so that's really where I want to end it. I'll leave it on this slide, but be happy to take any questions since I know we're at time.
Thank you, Nick. We do have a question here. Do property prediction models produce confidence intervals? If not, can you derive them from cross-validation?
Short answer is they can. So we use a variety of property methods depending on what we were training on. One thing I did not spend time talking about here is that we found that by working from our overall protein representation, we can train many of our property models with only a few hundred data points, not the thousands or millions that are known to be needed for deep learning. And so those are with different methods. Some of these do give us confidence limits, such as our blood-brain barrier penetrating models. For others, we can't do them for cross-validation. We also do some limited testing to develop those. And one of the things that we are doing concurrently with all of this is developing a set of benchmarks so we can have that sort of confidence limits to understand the quality of our predictions as well as our domains of applicability.
Okay, that's the only question I have. So I'd like to thank you, Nick. I really appreciate the presentation.
Okay, thank you, Brian. It was a pleasure. Thank you.
All right.